The wild type gene is changed, one amino acid change. So that's going forward, so to speak. Reverse mutation is, it already has a mutation, now the mutation has been corrected. A missense mutation is a forward mutation. Now you have nonsense mutation, which changes the codon to a stop codon. Now silent mutation is change in the codon but no change in the amino acid. So since we know that the genetic code is redundant and more than one codon can code for the same amino acid, in this case, if one nucleotide is changed, probably that new codon will still code for the same amino acid because the genetic code is redundant. In such a case, such a mutation is referred to as a silent mutation. So there is a change in the nucleotide, but there is no change in the amino acid. So that is a silent mutation, and that's because the genetic code is redundant. The genetic code, you know that more than one codon codes for the same amino acid. So such a mutation which changes the code but does not change the amino acid is a silent mutation. So look at this. There is no mutation normally DNA, RNA, protein synthesis. This is what we know is central dogma. Here, a missense mutation is where one nucleotide has been changed. So here instead of serine, you have leucine amino acid. So this is a missense mutation. Nonsense mutation is once again change in one nucleotide, but now that has become a stop codon. So no amino acid is synthesized here, and this is protein termination here. Silent mutation is one nucleotide has changed, but this UGC also codes for serine, just like UCA. UCA codes for serine, UCG codes for serine. So even though a nucleotide is changed here, there is no change in an amino acid. So that is referred to as a silent mutation. Now, more of them. Neutral mutation. A neutral mutation is a missense mutation that alters the amino acid sequence of a protein but does not change the function of the protein. Because sometimes when you have a mutation, the amino acid changes. So when amino acid change, the protein folding changes. Not always does the protein folding change, because the sequence of amino acids is what actually, you know what, amino acids actually have a pull for one another. They have attraction for one another, which actually leads to the folding of the polypeptide chain, and the protein folding gives its conformation activity. Sometimes, even though you have a mutation, even though one amino acid is changed, if in case this amino acid is not important for the protein folding, because not all amino acids are involved in protein folding. Some amino acids pull some other amino acids and the protein folds. In this case, let's say you have mutated, if there's a mutation that leads to an amino acid which is not important for protein folding, so even though you have an altered amino acid, the protein function is not lost. Such a mutation is referred to as a neutral mutation. Now you have loss of fate and gain of fate mutations. Loss of function mutation is where change in one amino acid will completely result in the protein losing its activity, because probably that amino acid was important for the protein to fold, or probably that amino acid was in the active site of an enzyme. Let's say a protein is an enzyme. Now there's a mutation in the active site such that one of the important amino acids has been mutated. Now the enzyme has become non-functional. This is referred to as loss of function, because the function of that protein is lost because of the change in one amino acid. Referred to as loss of function. Where is gain of function? Let's say there is one amino acid that's changed, all of a sudden, one nucleotide is changed, all of a sudden, which leads to the activation of a gene, and that gene is actually a transcription factor, for example. Now that transcription factor was not supposed to be expressed in those cells. Suddenly it's expressed because that one mutation has caused the transcription factor to be expressed. Now this transcription factor binds to the genes and leads to gene expression. All of a sudden, the cell probably assumes a different fate, just because you have one mutation which activated a gene that now is activating another gene. So such a mutation is referred to as gain of function. This gain of function causes the cell to produce a protein whose function is not normally present. So such a mutation is referred to as gain of function mutations. Now there are some mutations which are referred as conditional mutations. The mutation indeed has happened, but the gene is not activated until the temperatures have increased, or until there is more oxygen in the atmosphere, or until there is more sunlight, something like that. So some mutations which can be only active depending on the conditions or depending on the surroundings is referred to as a conditional mutation. See, for example, some conditional mutations affect the phenotype only at elevated temperatures, Still others are lethal. Some mutations can be lethal mutations as well. These mutations are the ones in which, sorry guys. It's a different sentence altogether. Okay, so conditional mutations where genes are, there's a mutation that affected the gene, but you don't see the effect of that mutation until the temperature increases, or until the oxygen content is more, or until the sunlight is more. So that mutation is referred as conditional mutation. There are other mutations called lethal mutations, because that one mutation will kill the organism itself, because that protein is probably very important for survival. So that is referred as lethal mutation. So we looked at lethal mutations, conditional mutations, loss of function, gain of function, neutral mutations. I've looked at silent mutations, nonsense mutations, missense mutations. Then we looked at forward mutations and reverse mutations. So these are 8 to 9 different terms that are pretty easy to remember. I mean, if you just follow the colors, you kind of get it. Now, other mutations going forward is suppressor mutation. What is suppressor mutation? A mutation in a gene that actually hides or suppresses the effect of another mutation. So let's say you have a mutation, right? There's a change in the gene which now causes a function to be lost or something to happen. Now there is a mutation in a second gene now, all of a sudden. This mutation, the second gene, is actually masking the effect of the first mutation. So now it all of a sudden looks like a wild type. Let me just jump the slide, it's easy for you to understand that way. So look at this here, this is Drosophila. We studied about, normally Drosophila have red eyes, and you know some mutations called it white eyes. So let's say for this example here, this is genes A and B express the red eyes. There is a mutation in gene A which causes the white eye. All of a sudden there is also a mutation in gene B which now makes the Drosophila have red eyes once again. So basically this second mutation is masking the effect of the first mutation. Such a mutation is referred to as suppressor mutations. Second mutation neutralizes the first mutation, let's put it that way. Okay? The second mutation neutralizes the first mutation. Now, if these second and first mutation are within the same gene itself, that is referred to as intragenic. Okay? I said one gene has an effect, mutation. The second gene somewhere now is kind of neutralizing the first one. I call it suppressor mutation. Now, if these first and second mutations are in the same gene, let's say two different nucleotides are mutated in the same gene. Now the second mutation I actually neutralize the first one. That is referred to as intragenic, because both the mutations are within the same gene. An intragenic suppressor mutation takes place in the same gene that contains the mutation being suppressed. Okay? See, the suppressor may change the second nucleotide in the same codon, altering the original mutation. So let us say, yeah, you know what, the slide is, it's easy. Okay, look at this here. Guys, okay, so this codon codes for leucine, you know that, right? One amino acid is changed, so the amino acid, so one nucleotide is changed, so the amino acid changed. Now, a second nucleotide in the same codon is changed, now it's back to leucine. You see that? So this is referred to as intragenic. So intragenic suppressor mutations. So intragenic suppressor mutation takes place in the same gene that contains the mutation that is being suppressed. So first is the mutation, you again change another code, another nucleotide within the same codon, for example, that puts back the amino acid back to place. So in the original mutation, for example, is one base deletion also, and then addition of a single base elsewhere in the gene will restore the former reading frame. This is also an example. So for example, here I quoted that if you change one nucleotide, the amino acid change, you change the second nucleotide in the same codon, the amino acid is back, using is back. Also likewise, let's say if you remove one nucleotide, what happens? The whole reading frame is shaped. Let's say you insert a different nucleotide in the same spot. What happens? The reading frame is restored. So this is also an example for intragenic suppressor mutations. The second one is intergenic suppressor mutations. The only difference is that in intergenic suppressor mutation, this actually occurs in genes other than the one being originally mutated. So previously I said first mutation in one gene, another nucleotide in the same codon is putting it back. Here I am saying there is one mutation in one gene. Now there is a second mutation in an entirely different gene which can actually neutralize the first gene. How can this actually happen? Let me jump to the slide, it's easy for you to understand. Then you can come back to the text here. So here, if you see this, there is, transcription lead to translation, right? Now, if you look at this here, one of these nucleotides here has been changed. So once you change the nucleotide, this has become a stop codon. If it's a stop codon, there is protein termination early. So this is the original length of the protein. Now you have a short protein because a mutation has changed the nucleotide in such a way that this has become a stop codon. Now if you have a stop codon in one gene, the protein expression is terminated. But however, if you take a second gene which codes for transfer RNA, right? Transfer RNAs carry amino acids. Now there are no transfer RNAs for stop codons. However, if you mutate one of the nucleotides for this transfer RNA anticodons, the base of the transfer RNA has anticodons which pair with the codons on the messenger RNA. We know that, right? Let's say one of the nucleotides in this transfer RNA base, which is this anticodons here shown as AUA, if they are mutated, they will still carry an amino acid, but they can now bind to the stop codon. Normally, there are no transfer RNAs which can bind to the stop codon, but here, since there is a gene mutation in the gene that codes for the transfer RNA, that now transfer RNA can bind to the stop codon. Now protein synthesis will continue once again. In this way, you have full length protein production. Guys, is that part clear or is it confusing? Yes, any questions on this one? Yes, no?
Is it important for the test?
That's a very good question, actually. That is the best question to ask. Yeah. Let's say no, you will not talk about it, so yes. So there's a mutation that leads to stop codon protein termination. But there's a second mutation in a gene for the transfer RNA. Transfer RNAs are the carriers of amino acids. If there's a mutation in the gene for the transfer RNA, the anticodons now all of a sudden they start binding to the stop codon here. Normally you will never see a situation with a stop codon here in red and you'll never see a transfer RNA sitting on it. But here it's sitting because this gene is a transfer RNA gene. It synthesizes a transfer RNA. But there's a mutation in this in such a way that the anticodons on the transfer RNA are complementary to the stop codon. And they bind and they also carry the same tyrosine amino acid just like shown here. Now that leads to protein synthesis once again. So this is referred to as intragenic suppression. In other words, intragenic suppression is the mutation in one gene is neutralized by a mutation in an entirely different gene. that is intergenic. Intragenic is where mutations happen in the same gene in two spots, which first is a mutation, second is the neutralizing mutation. So look at this here. They work by changing the way the messenger RNA is translated. Example, AAC, which is actually the messenger RNA, codes for leucine. This mutates to ATC, which is a stop codon, right? The stop codon mutation could be suppressed by a second mutation in a different gene that codes for the transfer RNA, which would result in a transfer RNA anticodon capable of pairing with the stop codon. Normally there are no transfer RNAs which pair with the stop codon, but here, since there is a mutation, the anticodon is now exactly complementary to the stop codon, so it binds on it, thereby protein synthesis continues. Okay? This is a slide giving you the important points that we have discussed: forward mutations, reverse mutation, missense, nonsense, silent, neutral mutations. In fact, this slide is actually more informative than the previous one. I did not go into in-frame deletion, expanding nucleotide repeats, or don't worry if I did not teach about that on the slide. I mean, there's so many slides. And this, guys, one thing I want to tell you before I go forward. If you go to slide 47 here, right? 48, I say extra slides, and then I have 20 more slides. Don't worry about these 20 slides. I say extra slides after 47. After 48 extra. So don't worry from 49 to 72. There are too many slides on this chapter. I have to cut them out, so I moved them to the bottom. So I will not be teaching it, and those slides will not be asked for the exam as well. Yes.
What part does it? I got it. Sit down.
Extra slides, right? So basically, the chapter is so big that some regions, some parts which I thought was not so much important, I moved them down, just to leave it out. But still, in the top 48 slides, 47 slides, I did cover a significant part which you guys can study and be ready for the exam. Okay. So we looked at all the different types of mutations. Now we look at what are the factors that cause mutations. All these are mutations, right? Nonsense mutations, silent mutations, whatever. So all these are different classifications of mutations that we look. But now the question is, what is actually causing these mutations? What are the factors that lead to mutations? So some of the factors are spontaneous and some are chemicals or radiation. Even though they use the word spontaneous mutations, which means mutations happen just by themselves, we all know that nothing really happens just by themselves. Something has to happen. So in this case, there is some mechanism that goes wrong within the cell and there is a mutation. Such mutations are referred to as spontaneous mutations. If the cell is functioning properly and if you still have mutations, that's because of chemicals or radiation. Basically, some chemicals can affect the gene. Some radiations like X-rays and gamma rays can also affect the nucleotide. So you have spontaneous mutations which are because of some defects in polymerase or some enzymes, and you have chemicals and radiations which are mutations caused by chemicals and radiation ions like X-rays and gamma rays. So we look at the spontaneous and the chemical and radiation factors that cause mutations. Okay? Okay. Venom is spread within the okay. Alright, you know what? Venom is spread when there is a mutation that is actually incorporated into the DNA. This mutation will continue on and on to the next generation because the DNA polymerase will read this mutation as a gene sequence and it will add the complementing nucleotide. So this text here in this chapter is kind of more generalized, so just read it. So look at this here. Okay, causes of spontaneous errors. Now we'll discuss the first. I said spontaneous mutations, then I said radiations, and I said chemicals. First, we'll look at spontaneous mutations and we'll see what are the reasons for spontaneous mutations. There are several reasons. One reason that can cause spontaneous mutations is actually crossing over. So in meiosis, we know that where there is exchange of genetic material between homologous chromosomes, there any error in crossing over, if the genes are not properly crossed over from one chromosome to the other, that could lead to spontaneous mutations. That is classified under spontaneous mutations. You also see something called strand slipping, where nucleotide strands form a small loop, and once the nucleotides form a small loop, what happens is they are not seen by the polymerase, and then these nucleotides are actually skipped, or sometimes the loop is formed in such a way that that nucleotide is inserted as an extra nucleotide. So if you look at this slide here, this is an example for spontaneous mutation, which shows how if there's any problems related to improper crossing over can lead to spontaneous mutation. This is one. The second one is here referred to strand slippage. Strand slippage is shown here, where you can see that one of the nucleotides has looped out. So you have you have 4 A's here, but you only see 3 A's here, base pair, and one of the A's actually looped out. In such a situation, this results in the addition of one nucleotide on the new strand. So this is one way. The other way is that template strand loops out in such a way that the resulting in the omission of the nucleotide in the new strand. So this kind of strand slippage can lead to insertions or deletions within the new strand. And these are also classified under spontaneous mutations. So strand slippage is under spontaneous mutations. Crossing over is also classified under spontaneous mutations. Now, there's something called, another example for spontaneous mutations is depurination, which means loss of a purine base. When you lose a purine base, that DNA is now referred to as apurinic. So when you use the alphabet A before anything, which means it's without purine. Apyrimidinic means without pyrimidines. So apurinic is nothing but loss of purines. Usually an incorrect nucleotide is incorporated. So once you lose this purines, what happens is that loss of a purine is actually replaced by the enzymes, and usually for some reason adenine is incorporated. Now that adenine is now incorporated into the DNA in such a way that during the next cycle of replication, that the DNA polymerase reads this adenine and adds a thymine on the second strand. So this mutation has now, in more or less in a way, has become prominent. Don't worry, it's on the next slide, that information, which I deleted. So look at this depurination. Here, if you look at it, this purine base here, for example, guanine is an example for purine, right? Guanine is lost. Once guanine is lost, what happens is the polymerase realizes that it sees a sugar molecule, but it does not see a nucleotide. So instead of, for some reason, instead of putting back a guanine, it adds adenine. And that adenine, when incorporated into the DNA, is passed on from generation to generation. So that mutation has now become permanent within the DNA. So this concept where purine is lost is called depurination. Depurination is nothing but loss of a purine base. So this depurination results because of the covalent bond which breaks between the sugar molecule and the purine base. Usually an incorrect nucleotide, most often an adenine is added. Why is adenine added if guanine is lost? It is not really known, but I think it's related to the defect in the enzyme. The enzyme sometimes cannot replace the right nucleotides. So the other classification for spontaneous mutations is deamination, where here, if you look at it, sometimes an amine group is actually removed from one of these bases. So cytosine actually has an amine group. If amine group is actually lost from cytosine, now cytosine all of a sudden becomes uracil, because the difference between cytosine and uracil is that cytosine has a amine group and uracil does not have an amine group. Loss of an amine group or suddenly makes the cytosine into an uracil. This is actually referred to as deamination. Here, don't know very good example, but look at this. So this is good enough. So deamination is the loss of an amine group, and once you lose the amine group, cytosine all of a sudden becomes uracil, because this is the structure of uracil. Uracil does not have an amine group and cytosine has it. Now, if there is deamination leading to loss of amine group, the base is automatically changed. Now, we come to, after looking at spontaneous mutation, which are because of some defects in enzymes or because of defects in loss of some groups, we look at something called chemically induced mutations. So chemicals and radiations can also cause mutations. So some chemicals, let's look at some examples for chemicals and radiations. So one example is base analogs. There are some bases which are not nucleotide bases, but they look like nucleotide bases. And one good example is 5-bromouracil. This is not uracil, it's called 5-bromouracil. 5-bromouracil looks like thymine. It is an analog for thymine. We used to use BRD assays. Normally in the lab we do some experiment called proliferation assays, where you want to see how a cell is proliferating. For those experiments, we used to inject 5-bromouracil into mice. So this 5-bromouracil looks like thymine. So what happens is, DNA polymerase will think that 5-bromouracil is thymine, and it will add that thymine into the 5-bromouracil into the DNA. So look at this. This is the structure of thymine. This is the structure of 5-bromouracil. Both of them look almost the same. And once 5-bromouracil is actually incorporated into the DNA, it actually can base pair, it can form complementary strands with adenine, it can also form complementary strands with guanine. So this is a base analog. In other words, it is not the natural base, but it can mimic thymine. Once it gets into the DNA, it can base pair with adenine or guanine. So this is referred to as base analog. Base analogs are chemicals which look like nucleotides, but they are not nucleotides. Okay, the other one is alkylating agents. Alkylating agents, what they do is, they usually donate their methyl group or the ethyl group. These methyl group and ethyl groups, which are donated by the alkylating agents, they are now added to the nucleotides with the help of the enzyme called with the help of ethyl methyl sulfonate. So this has the ability to add the methyl group or the ethyl group to the bases. Once you add methyl group or ethyl group to the nucleotide bases, the nucleotide bases now have a different structure, but the DNA can actually accidentally add these to the this to the growing strand of the DNA. So alkylating agents are also chemicals that can actually modify the nucleotides, leading and kind of cause these modified nucleotides to be incorporated into the DNA with the help of DNA polymerases. So deaminating chemicals. I previously said that amino groups actually are removed from the DNA, which I classified under spontaneous mutations. But then I did not mention what causes deamination. So nitrous oxide can deaminate cytosine, creating uracil. So here, nitrous oxide is acting as a chemical which changes cytosine to uracil by removing the amino group. So nitrous oxide is an example for deaminating chemicals. Likewise, hydroxylamine. This actually adds one hydroxyl group to cytosine, converting the cytosine into hydroxyl aminocytosine. So these are different chemicals. So guys, if you look at it, to put it simply, base analogs alkylating agents, deaminating agents are examples for chemicals which can alter the bases by adding or removing different groups. Okay, look at this here. This is example for alkylation, where guanine is linked with, when it's alkylated with a group, it becomes O6 ethyl guanine. This guanine can, this O6 ethyl guanine can now base pair with thymine. Normally guanine only base pairs with cytosine, but this is not guanine, it's O6 ethyl guanine, it pairs with thymine now. Other example is deamination, when you lose an amine group, cytosine all of a sudden becomes uracil. Okay? The other one is hydroxylation, where cytosine, where hydroxyl group is added to cytosine, and this hydroxyl aminocytosine can now base pair with adenine. All these are examples for chemicals that actually can modify DNA bases. Now we come to radiation. So when you speak about radiation, we look at X-rays, gamma rays, and cosmic rays. All these radiations, all these high energy particles can actually cause covalent bonds between two different nucleotides, which actually is a way to say that radiation can also cause mutations. So look at this. Here you see that pyrimidine bases can actually absorb the UV light, which causes chemical bonds to form between adjacent pyrimidine molecules on the same strand of DNA, creating pyrimidine dimers. So look at this here. Here you have two pyrimidine bases, which is thymine and thymine. Now, if they are hit with UV light, that energy absorbed from the UV light actually helps create a covalent bond between the two pyrimidine bases here, referred to as thymine. Now you have a dimer formed here, so there's actually a different kind of bonding between this thymine and thymine, which is to show an example how radiations can actually affect the DNA. That's clear, guys? So this is how radiations can have an effect on DNA. Now, after looking at chemicals that can actually cause modifications in DNA, and looking at radiation that can cause modifications in DNA, now we look at transposable elements. Remember, we spoke about transposable elements in the previous chapter. We said transposable elements are nothing but jumping genes. Genes that move from one chromosome to the other chromosome. In other words, let's say there is a gene on chromosome 1, it copies itself, it makes a second copy of itself, and it now moves to a different chromosome. When this gene moves to a different chromosome, it could now land in between a different gene, destroying the second gene. Get that? So one gene has copied itself, it made a second copy of itself, it now has moved on to a different chromosome. So when it inserts itself into a different chromosome, it inserts itself obviously probably into another gene, thereby destroying the second gene. This is also an example to show how jumping genes, referred to as transposable elements, can actually cause mutations. So DNA sequences that can move about in the genome often cause mutations, and these transposable elements are called movable genes. Controlling genes are also called jumping genes. Because on either side of these genes, you have some direct repeats, or basically some short nucleotides, which can be cleaved, and that whole, now once these short nucleotides on either side of these jumping genes are cleaved, the whole gene now moves from one chromosome to the other chromosome by copying itself. These flanking repeats are created when staggered cuts are made. These staggered cuts are actually made by transposase, an enzyme. There are enzymes that cut the DNA in these direct repeats, and now these genes have copied themselves, and they actually move from one chromosome to the other chromosomes. Transposable elements are actually found in prokaryotes and also in eukaryotes like us, like human beings. So how does the whole concept of jumping genes actually work? There are 3 steps. One is staggered breaks, basically DNA on either side of the gene is actually broken. Now the transposon element, which is actually the gene, is joined to single strand ends of the target DNA, and then once it moves to a new location, DNA is replicated at the single strand gaps, and then these gaps are actually sealed. So there are cuts made on the DNA with the help of the enzyme called transposase. Now the gene is actually moved to a different location, and there is a new cut made